I'm going to be talking about data visualization in proteomics. And as we heard this morning from Sean that even one protein is a beast in itself, then imagine the whole proteome, and it's going to be a monster, I think. Because of the complexity of the proteome, it's really hard to define. It's not like in genomics where the reference genome is more clear. Multiple protein isoforms, PTMs. It varies in time and space, and there's an extremely high dynamic range from the lowest expressed to the highest expressed protein. It's like 6 orders of magnitude. So basically, there's possibly an infinite number of different protein species. So defining the proteome is, of course, the entirety of all expressed proteins, but a more workable definition is that to identify and quantify at least one protein species from every expressed genomic locus. So how do people measure the entire proteome? Well, people have done it one by one with antibodies, or more recently with mass spectrometry. Some people have also really done high throughput measurements using fluorescent tagging of individual proteins, such as Uri Alon's work, where he's tagging up to 1,000 different proteins and tracking them during cell cycle and response to drug. There's also been great developments in single-cell proteomics. For instance, Gary Nolan, who's merging flow cytometry and mass spectrometry to measure up to around 50 different protein species in single cells. I'm going to be focusing on the first 2 methods here and presenting some of the work of our groups in CBio Portal and a tool called NetForest. But before we go into that, I'd like to talk a bit about what we think is important for data visualization. This is just a few characteristics. But for proteomic repositories and tools, it's of course important to incorporate multiple types of data, not only protein data, but as many different types as you can get. And you have to organize it in a systematic manner and use and classify your data. And this is, of course, not new. 500 years ago, Ulrich Pinder made this urine wheel where he classified urine based on smell, color, taste, and linked it to different diseases as a diagnostic tool. So it's important to integrate these many types of data and different data modalities and classify them. But of course, it's also important that we do some kind of hypothesis testing. We need to discover something with our tools. So we need to be able to model the data. And finally, it's important we share with the community, as we've seen many of the tools presented earlier here. So starting with the antibody-based proteomics, what we have been mainly involved in are these reverse phase protein arrays where you simply spot lysate from your cells or your tissue of interest and spot it on these chips, and you can have up to 1,000 different samples per slide. And we've been working with Gordon Mills at MD Anderson in Texas, and they have a repertoire of around 150 antibodies, and some of these are recognized phosphorylation sites. And you really need very little sample. So this is great if you have a lot of samples and want to measure some proteins of interest. And these are, of course, selected for certain cancer proteins. So last year, I presented a little bit our work in that, where we're basically, in this case, I'm working with liposarcoma, doing different perturbations and combinations of drugs, and then measuring the response of around 50 or so phosphorylation changes. And what we then try to do here is derive network models directly from the data itself and try to link each measured node, each measured protein in a quantitative manner in a network. And we typically generate up to 100 or thousands different models that represent the data. So we had a challenge of visualizing this many different models onto a set of nodes. So we tried to incorporate multiple edges between nodes. But since this is not about networks, we're going to hear Scooter talk about that in a moment. I'm not going to focus on this, but I just wanted to talk about our work in TCGA consortium. So we're also involved in the Cancer Genome Atlas, and here again, Gordon Mills in Texas their group have profiled many samples now with the reverse phase protein arrays, and we've incorporated this into the CBio Cancer Genomics portal. So if we take a look at that, we've now included around 30 different studies, and this is, of course, ongoing, of different cancer types, which is profiled by many different genomic methods, but also RPPA, as you can see here. So out of these 10,000 tumor samples that are in this data bank, about half of these have proteomic profiles. So this is actually a really great resource for digging into protein changes across different cancers. So, for example, if you're interested in EGFR, epidermal growth factor receptor, and you can get an overview here of the different cancers where it is mutated or amplified. And in this case, you can see that glioblastoma is one of the brain diseases where EGFR is highly amplified and mutated. So if you go further and look into just GBM and see what the changes to the EGF receptor is, then we get these small oncoprints that we call them, basically a small multiple of all your patients, and you can get an overview of what percentage of your patients have altered EGFR. But as this is about proteomics, you can, of course, also see what protein changes there are linked to the changes in EGFR. So very obviously, the EGFR phosphorylation of EGFR, in this case, these tyrosine phosphorylations, they're positively linked. If you click on the buttons here, you can then see that the altered set, so these 50% or so that are altered on a genomic level, also have high expression of phospho-EGF. Not surprisingly that EGFR is overactive in these tumors. But what you also can see is here that the bottom one, the RAF1, which is actually downstream of the canonical MAP kinase pathway of EGFR, is unexpectedly upregulated in the unaltered set of samples here. So this is counterintuitive, but of course, RAF1 is altered. It has multiple upstream pathway activators. So in GBM, for instance, we know that NF1, a tumor suppressor gene, is often either deleted or mutated, and which results in a loss of function. So now in this query, just for the sake of demonstrating our database, I put in NF1 here, but also the RAF1 phosphorylation site here. So any protein that's highly upregulated in RAF1, we can now query that. And then you get these oncoprints again, and now a pattern starts to emerge that you can see that most patients have these EGFR mutations, but certainly also some have the NF1 loss of function, and even RAF is increased in some patients. So there are exceptions, but what you can conclude or maybe see from such data is that there's some kind of mutual exclusivity going on here. You need either an alteration in EGFR or an alteration in RAF1 or an alteration in NF1. And there is a co-occurrence between NF1 deletions and RAF1 upregulation, because NF1 is activating RAS, but since it's lost, so it's a disinhibition. So there are also tests that you can make here for mutual exclusivity, and in this case, we can see that there is some kind of mutual exclusivity going on between NF1 and EGFR. So basically, people can click all these different tabs, get network information, get survival information based on the query that you input. And Gideon, who's in the audience here, will give a live demonstration on his poster tomorrow. So we now, of course, want to use the mass spec-based method instead of the RPPA, because RPPA, although it's powerful on a sample size level, then there are variable specificities of the antibodies. So for the tumor projects, we're collaborating with Rudy Aebersold, who is then doing targeted mass spectrometry of the tumor samples. And we have another ongoing project with Forrest White on the other side of the street, where we want to do tumor biopsies before and after treatment and compare the proteomic profiles of these tumor biopsies. So, but this is all in the making, and we unfortunately don't have any data yet. But our other long-standing collaborators are the Matthias Mann group, and what they typically do is the so-called shotgun proteomics, where you want to try to map as much as possible of the proteome. And so the setup for the targeted mass spec and the shotgun mass spec is more or less the same. There's some different in the instruments, but you want to digest your proteins into peptides and then shoot them through the mass spectrometer, and then ultimately you can even sequence your peptides and then map it back to the proteins. So Matthias Mann's group has done a huge, tremendous amount in making this better and better and better, and they are at a stage right now where reports from their group show that they can, in a 4-hour run, identify 4,000 proteins from the yeast proteome, which basically is all proteins that are expressed in the yeast. And so this corresponds to the time that it would take to do a single Western blot. So comparing this with other methods like tab tagging and GFP tagging, they can see that mass spec in a 4-hour run actually covers much of the same space, if not even more, and from 4 orders of magnitude from the lowest expressed molecules to the highest. So basically, not all proteins, not all genes, all the open reading frames does not give rise to expressed proteins. So they're reaching the limit of what and measuring of what the entire proteome. So together with Lars, we've analyzed some of their data in collaborations a couple of years back, where we were mapping around 20,000 phosphorylation sites during the cell cycle in HeLa cells. And you can see this heat map here, basically the different samples from cells were synchronized and taken in the different phases of the cell cycle, and then we did a large-scale phosphoproteomic analysis of this. And you can see some patterns emerging of, here are some of the, actually almost half of the phosphosites change more than twofold during the cell cycle. So you can see some patterns of which proteins are phosphorylated at what stages in the cell cycle. So to make an artistic representation of this, I just took a part of these cycling phosphosites and wrapped it around a globe to sort of say we're doing a global cell cycle phosphoproteomics. So all these massive amount of data are stored in databases, and here's a database from the Mann group that collects all these phosphorylation sites that they identify in many different species. And again, if you're interested in EGFR and a certain site on EGFR, you can see how this site fares during different experimental conditions, including EGF stimulation or in the cell cycle. But it's not only phosphorylation that's in such databases. Now also other PTMs is coming in. But how do you make sense of all this data? So again, in a close collaboration with Lars, we wanted to make a tool that could take a list of high-quality phosphorylation sites that were identified in mass spectrometry and then try to relate it to a biological context through mapping it to signaling networks, or at least also just trying to map which kinase phosphorylates what site. Are there any of these tyrosine phosphorylations that attract SH2 domain containing proteins to further propagate the signal? So what we did was we first started to collect all the knowledge that was in databases and in the literature of kinase and substrate pairs. So these were, individual experiments where people had shown a certain site had been phosphorylated by a specific kinase. And then we mapped this onto the 500 different kinases of the human genome here. And this is clustered by the sequence similarity of the kinase domain. And then the bars here are indicates where we could find data where we could link a certain kinase to a certain substrate. But of course, you don't always know if it's a specific kinase that phosphorylates this site or it's a group of kinases. So what we did was we made these bubbles here for sites where we only knew if it was a certain group that phosphorylated this. And this is, by the way, visualized with the ITOL tree that Letonich talked about a couple of years ago at VIZBI. Also for the SH2 domains, we did the same exercise. And we also, in blue here, we have additional data which are either kinases or SH2 domains that had been profiled in vitro for their sequence recognition motif. So we also mapped that onto the trees just to get an overview of what we had. But such visualization actually also helped us in the design of the computational method, because we thought, or Lars thought, that if you have a specific site, a group of kinases could phosphorylate this. So in cases where we didn't have that knowledge, we could make group predictors, meaning that if the same kinase has or two different kinases have a very similar sequence, they probably phosphorylate the same data. So in this way, we could use this tree as a structure for our computational method. So what we wanted to do was take these individual sites, so sites and kinase relationships, the whole tree, the kinome tree, and these in vitro assays that people had profiled each kinase for their sequence specificity, and then plug this whole into a pipeline that automatically would train artificial neural networks to recognize and predict new kinase-specific phosphorylation sites, and do automatic benchmarking and automatic selection, and in the end up with a set of robust classifiers that are also a non-redundant set of classifiers. So we did that and mapped it back to the tree here, and so now the orange here are the artificial neural networks, the different models that we generated from the data to being able to predict new sites for a given kinase. Or we used position-specific scoring matrices for the in vitro data. So now we can see that we cover about half or a little less than half of the kinome, but most of the SH2 domains in this manner. So now we have a prediction pipeline and a prediction tool for given sites and in order to classify what upstream kinases actually phosphorylates this site. It can also be used as a simple lookup atlas, and here we made a poster so bench biologists can go in and check out their favorite kinases and see what they recognize. In this case, EGFR doesn't have a strong motif, but there is some preference for hydrophobic amino acids in black here and some acidic amino acids in red here. But with other contextual information, we can actually increase the prediction. So coming back to the cell cycle data, then this is good with a heat map, but we thought that maybe we can use this tool, NetForest tool, to do a more kinase-centric analysis of this. So what we did was we took all these cycling phosphosites and predicted whether there were some enriched motifs in the different kinase groups. And as expected, most kinases are not involved in the cell cycle, so this is black here. But there are some ones that recognize and have important roles in the cell cycle, for instance, the CDK kinases. So this is a different view of the same data, and something emerges like there's possibly a role for ATM and ATR kinase. Maybe it's because of the link between DNA damage and during DNA replication. So what we're doing now in collaboration with Rune Linding is we put in protein association networks to even get a better to get better predictions. But not only that, we can now start to make signaling network models based on input set of phosphorylation sites. So this is ongoing work, and we're expanding this to different organisms in the near future. But since this is, again, not about networks, I'm not going to show more about that. I'm actually going to end off here by saying what we think are some of the challenges in proteomic data visualization. So specifically for proteomics, it's of course important how comprehensive are you if you're analyzing the entire proteome. And rather than maybe being focused on specific types of data like proteomic data versus genomic data, then one way that is important is that we could be focused on a biological topic like cancer, for instance, exemplified with the CBio portal, that we don't really care if our data is RNA-seq or genomics or proteomics, as long as we can learn something new about what we're interested in. Another thing is the systematicness of these methods, and for proteomics specifically, it's important that all these analysis pipelines are automated and standardized, because there's so many different types of processing tools out there that really a standardization is necessary. And in order to show all this data, I mean, we need these small multiples is one way of doing this. And the proteomic world also tends to just produce big lists, so we really need to be able to make tools that are predictive and can model the data and hopefully provide new insights. So you shouldn't just be able to put in one protein, you should be able to put in multiple proteins or genes and then see what the correlations are. And finally, proteomics sometimes tends to be closed on itself, so it's really important that we share these tools and databases. So for future directions, I think visualizing cell-cell communication between multiple cell types is going to be really important. And together with another postdoc in the lab, we've developed this new technique where we can actually selectively label the proteome of two different cell types growing together in co-culture by modifying a version of SciLac. So we can now directly lyse co-cultured cells and see what proteins come from which cell types. And this is extremely important if you want to track the cell of origin of proteins, and also if you want to investigate how, for instance, stroma cells like macrophages influence tumor cells and how they influence drug resistance responses. So really, we think we have a new cool method where we address a need of being able to use high throughput techniques to identify cell of origin of proteins. And this is, of course, important, especially for secreted proteins where an antibody, even though you don't know where the cell of origin is, this method can do this. So if any of you are interested in cell-cell communication and the problems of visualizing this, I'd like to talk with you during this conference. Another emerging field is going to be the personal proteomic visualization, and of course, the genomics are always a little ahead of proteomics, but here's an example from the VIZBI portal, again, where there's a patient viewer where each patient, all the alterations and mutations, you can click on these and get more information, but you can also find suggestions of drug treatment and the clinical trials that are going on. So this is one step closer to the personal treatment idea. And finally, we're also going to have 1,000 protomes soon, so we need to think about how to visualize that. I want to thank Gideon, who's in the audience here, for the work. These people have been working on the VIZBI portal. This is my supervisor. And I want to thank Lars and Heiko. Heiko is also in the audience, and our collaborators at the University of Copenhagen, and my former supervisors at the Technical University of Denmark. Thanks.